Hey everyone, Dr. Steve here. You know it as well as I do, but most students need a B or better in A&P. But did you know that students are doing that all over the US using my Anatomy and Physiology Digital Suite as a low-cost alternative to expensive textbooks? Digital Suite delivers the key concepts of A&P with short, assignable tutor videos and a bulleted narrative study guide that not only explain the concepts of A&P, they show them happening. Combined with state-of-the-art dissection and physiology lab simulations and a vast question bank for quizzes, homework assignments, and exams, Digital Suite meets students where they are and gives them the tools to master the content. It's the perfect supplement to an OER textbook or just on its own. If you're an A&P instructor, contact your McGraw-Hill representative today and ask about Anatomy and Physiology Digital Suite. If you're a student, tell your instructor about it. There's a link to Digital Suite in the show notes of this episode. Get your B or better in A&P with A&P Digital Suite.
Happy holidays! Want to give your host a gift? Consider subscribing, rating, and reviewing the show this holiday season. It really helps the show grow. From all of us at Believe, have a Merry Christmas, everyone, and a Happy Holiday.
Hey everyone, welcome to episode 2 of my podcast, Anatomy and Physiology Bit by Bit. I'm your host, Dr. Steve Sullivan, and I'm still in a hotel room in Lexington, Kentucky. I just got done talking to a lot of really great colleagues down at Bluegrass Community and Technical College here. There's a lot of excellent A&P instructors here that are doing some really cool things with their students. They're getting ready to use a lot of digital technology to help their students learn, whether it's face-to-face, online, or hybrid. So it's been a really good experience here. I've had a really good time. Also, Lexington's really nice. Lots of horses, lots of open land. I found a good sushi place and a good Indian restaurant. Like I said, it's been a good time. It's not my first time here in Lexington. Years ago, I was here to speak with the anatomy faculty at the University of Kentucky Medical School. But this time, I got to connect with a couple of colleagues that I look forward to working with on my current publishing project. And I'm really excited for what they're going to do moving forward, helping students learn and become great clinicians. Speaking of clinicians, today's topics are so important for anyone studying to be a healthcare practitioner. Whether it's doctor, nurse, physician assistant, radiology tech, physical or occupational therapy, you name it. You need to understand life and homeostasis. There are essential characteristics that separate living things from non-living things, and those living things need to make sure they can survive in their environment. When working adequately, the process that ensures that survival is homeostasis. So let's get started with today's topics, beginning with the essential characteristics of life. When we talk about humans as organisms, we're really putting ourselves in the same category as bacteria, oak trees, and fruit flies. That can feel a bit humbling, but it's incredibly useful. It lets us use the same biological principles to understand a febrile toddler in the ER, a patient on a ventilator in the ICU, and a yeast cell dividing in a culture dish. At the heart of that commonality is one big idea: life. In school, it's easy to jump straight to disease like heart failure, chronic obstructive pulmonary disease, diabetes, without asking a more basic question: what exactly are we trying to preserve when we save a life? Today's story is about two tightly linked ideas. 1, the essential characteristics of life. 2, the homeostatic mechanisms that protect those characteristics from moment to moment. Think of this as the operating system of the body that your future clinical knowledge will run on. So what does it mean to be alive? From a biological perspective, something is alive if it consistently exhibits a set of properties, behaviors, and capabilities that distinguish living organisms from non-living matter. In this framework, there are 6 essential characteristics of life. 1, organization. 2, metabolism. 3, responsiveness. 4, growth. 5, development. And 6, reproduction. We'll walk through each of these not as abstract definitions, but as things you'll actually see as a healthcare practitioner someday, often in dramatic ways when they fail. We'll start with organization. from atoms to organ systems. Organization refers to the specific interrelationships among the parts of an organism and how those parts interact to perform functions. Structure and function are inseparable. That mantra from Anatomy Lab is the essence of biological organization. We talked about this in episode 1. You can think of organization in levels, from simplest to most complex. Chemical, atoms and molecules, water, ions, proteins, lipids, nucleic acids, for example. Cellular, organelles and cells, the basic units of life. Tissue, groups of similar cells with a common function, epithelial, connective, muscle, nervous, for example. Organ, 2 or more tissue types working together, like the heart, liver, kidney, skin. Organ system, groups of organs performing a major function, like the cardiovascular system, the respiratory system, the nervous system. And organism, the whole being. For us, it's the human being. This is all review, and I'm not going to get into a lot of detail because we covered this in the first episode. In a healthy person, these levels are exquisitely coordinated. An osteocyte, which is a bone cell, a neuron, which is a nerve cell, an hepatocyte, which is a liver cell. Each is specialized, but none can survive indefinitely in isolation. They are organized into tissues and organs that depend on each other. Imagine someday you're working in the intensive care unit of a hospital. A patient has septic shock, meaning an infection in the blood is causing them to shut down. They require multiple vasopressors, which increase blood pressure, mechanical ventilation to breathe, and dialysis to support kidneys that aren't functioning. The chart says multi-organ failure, but what's really happening is a loss of organization. The blood's smallest vessels, the capillaries, are leaking, and the arteries are dilated. This means the tissues aren't getting enough blood. The kidneys fail to regulate electrolytes and fluid volume. The lungs can't oxygenate blood, and the liver can't clear toxins. The organs are physically present, but their integrated function as a system is collapsing. It's a grim but powerful reminder that life isn't just having the parts, it's the organized interaction of those parts. Number 2 is metabolism, the chemistry of staying alive. Metabolism is the sum of all chemical reactions taking place in the cells and internal environment of an organism. It's not just burning calories, it's every reaction that uses energy, transforms molecules, or builds cellular structures. We often divide metabolism into 2 broad categories. Catabolism, which is breaking larger molecules into smaller ones, usually releasing energy, and anabolism, building larger molecules from smaller ones, usually requiring energy. When you eat, your digestive system uses specialized proteins called enzymes to break down carbohydrates, proteins, and fats into smaller units. That's catabolism. The resulting glucose, amino acids, and fatty acids are absorbed and used as fuel for cellular activity, which we use in the form of adenosine triphosphate, or ATP, and raw materials for building new molecules, lipids for the plasma membrane, structural proteins to build connective tissues and muscles, nucleotides to build DNA and RNA, for example. Cells then use ATP to drive anabolic processes, synthesizing proteins, repairing membranes, replicating DNA, and reshaping the cytoskeletons of our cells. A particularly vivid example of metabolism and structure comes from immune cells. Some white blood cells can dramatically change their shape, extending pseudopods to surround and engulf a bacterium. This shape-shifting depends on energy-driven rearrangements of the cytoskeleton. Metabolism literally driving form and function. Picture a patient with diabetic ketoacidosis, or DKA. This means that a lack of insulin is causing the body to break down fats for energy instead of using glucose. The ketones that result cause acidity and can lead to severe health issues. Their breathing is rapid and shallow, which is called tachypnea, they're dehydrated, and the pH of their body's fluids is too acidic. At its core, diabetic ketoacidosis is a massive metabolic problem. In the absence of effective insulin signaling, cells can't use glucose efficiently, so they switch to fat breakdown, generating ketone bodies and overwhelming the body's acid-base balance. You're giving fluids, insulin, and electrolytes, but conceptually, you're trying to restore normal metabolism, the seamless coupling of catabolism and anabolism that life depends on. Metabolism underlies every other characteristic of life. Without energy and chemical reactions, there is no responsiveness, there's no growth, there's no development, there's no reproduction. And those four are the next essential characteristics of life. The number 3, responsiveness, sensing and responding to change. Responsiveness is an organism's ability to sense changes in its internal or external environment and adjust to those changes. It's the biological equivalent of situational awareness. This happens at multiple levels. Cells can detect changes in their local environment, for example, ion concentrations, growth factors, mechanical stress. And when they detect those changes, they can alter gene expression or behavior. Tissues and organs contain specialized receptors, thermoreceptors for temperature, baroreceptors for pressure, chemoreceptors for chemical signals, and nociceptors which detect tissue damage. The nervous and endocrine systems provide the primary mechanisms of cell-to-cell communication, coordinating responses across the body. Responses include behaviors like moving toward food or water, withdrawing from a painful stimulus, or adjusting physiological variables to maintain a stable internal environment. A simple example. When external temperature rises, thermoreceptors in the skin and core sense the change. The brain responds by stimulating sweat glands to produce sweat, and by adjusting blood flow to the skin. Evaporation of sweat and increased cutaneous blood flow help lower body temperature back toward normal. We're going to talk about this in more detail later on with a different example. Think about the last time you accidentally touched something hot. Before you consciously registered hot, your hand was already pulling away. That rapid withdrawal reflex involved receptors in the skin, spinal cord circuits, and motor neurons activating muscles, all in a fraction of a second. That's responsiveness in action. At the clinical level, responsiveness can be striking. Someone who is allergic encounters a peanut protein or bee venom. The immune system's antibodies trigger cells to release large amounts of histamine, leading to a drop in blood pressure from vasodilation and constriction of the lungs' airways. The body is responding very vigorously to a perceived threat, but in a way that endangers the organism. Understanding responsiveness helps explain both normal protective reflexes and pathological overreactions. Number 4 is growth, getting bigger and more complex. Growth is an increase in the size or number of cells leading to an enlargement of all or part of an organism. There's 3 common ways this occurs. 1 is hypertrophy. Cells get larger. 2 is hyperplasia. Cells increase in number. And the third one happens in some tissues where growth happens by increasing the amount of extracellular matrix that's around the cells. In skeletal muscle, for example, resistance training stimulates protein synthesis and the addition of the contractile units inside a muscle cell. When that happens, individual cells, called myofibers, grow larger. That's hypertrophy. In bone, growth involves both increased cell number and the deposition of mineralized bone matrix around those cells. That's the hard, calcified part of bone tissue. In a clinical setting, growth is something you'll literally plot on a chart. Pediatricians monitor growth curves, tracking height, weight, and head circumference over time. Deviations from normal growth patterns can signal endocrine disorders, malnutrition, chronic disease, or genetic conditions. Sometimes in pediatrics, you might see a child who has, quote, fallen off the curve. Weight and height percentile steadily dropping over several visits. You start thinking about causes: inadequate caloric intake, malabsorption, chronic illness, hormonal abnormalities. Behind that chart is a simple question: why is this child's normal expected growth, an essential characteristic of life, not happening? At the other end of the spectrum, pathologic growth, like tumor formation, is also built on the fundamental capability of cells to grow and divide. Life's ability to grow is normally tightly regulated. When that regulation fails, pathology tends to follow. Number 5 is development. This is change over a lifetime. Development includes all the changes an organism undergoes from fertilization to death. Growth is part of development, but not the whole story. Development also involves differentiation, which means cells changing from an immature generalized state to a mature specialized state. And morphogenesis, changes in the shape of tissues, organs, and the entire organism itself. Right after fertilization, the resulting cell from the sperm fertilizing the egg is called a zygote. The zygote divides to form a ball of relatively similar cells. As development proceeds, these cells differentiate or develop into specific lineages. Some become neurons, others become muscle cells, still others become liver cells, which we call hepatocytes, and so on. Their structure and function diverge as they specialize. These differentiated cells organize into tissues and organs, and through morphogenesis, the body takes shape. Limbs bud and elongate, the heart loops, the brain forms distinct regions. The most dramatic developmental changes occur before birth, but development continues afterward. Teeth erupt, the brain matures, secondary sex characteristics appear at puberty, and the process of aging unfolds later in life. If you spend time in an obstetrics clinic or a neonatal intensive care unit, you'll see developmental biology made visible. A premature infant has underdeveloped lungs, an immature immune system, and limited fat stores. Their difficulty maintaining body temperature, coordinating sucking and swallowing, and resisting infection all reflect developmental stages that normally would have finished before birth. Someday you may learn about congenital malformations like congenital heart defects or neural tube defects. You're really studying disruptions of morphogenesis and differentiation. And finally, the 6th essential characteristic of life is reproduction, continuity from cell to species. Reproduction is the formation of new cells or new organisms. It operates on 2 levels. Cellular reproduction, one cell divides to form 2, allowing for tissue growth, repair, and turnover, and organismal reproduction, the generation of offspring, passing genes to the next generation. In your own body, reproduction is happening constantly. Skin cells divide to replace those shed at the surface. Cells lining the gastrointestinal tract turn over rapidly. Stem cells in the bone marrow produce new red blood cells, white blood cells, and platelets daily. Without continuous cellular reproduction, tissues would wear out and fail. Organismal reproduction is how we produce offspring. In humans, we do it via the formation of gametes, which are sperm and egg cells, and that results in fertilization. This ensures continuity of the species. From a medical standpoint, this shows up in fertility clinics, obstetrics, and counseling patients about diseases offspring can inherit from their parents. Consider chemotherapy. Many chemotherapy drugs target rapidly dividing cells. That's useful for killing cancer cells, but it also affects other proliferative tissues like bone marrow, the cells of the gastrointestinal lining, hair follicles, and cells that produce gametes. Patients develop anemia, infections, inflammation of the intestinal wall, hair loss, and sometimes infertility because the chemotherapy interferes with normal cellular reproduction, a fundamental characteristic of life. Okay, now that we've talked about what living organisms are capable of, we need to ask, how do those capabilities stay stable enough to be useful? Your cells are picky. Their enzymes work within narrow temperatures and pH ranges. Ion gradients across membranes must be tightly controlled. Energy supply and waste removal have to be balanced. The The process of maintaining a relatively stable internal environment is called homeostasis. So let's talk about the internal environment of our bodies and its variables. Homeostasis is the existence and maintenance of a relatively constant internal environment in the body. That phrase, relatively constant, is important because it's not perfectly fixed like a straight line. It's more like a controlled wiggle around a set point. As you go through your day, external conditions change. Temperature, activity level, emotional stress, nutrient intake. These changes influence internal conditions like body temperature, blood volume, blood pressure, concentrations of ions like sodium, potassium, and calcium, blood glucose, pH of body fluids, and oxygen and carbon dioxide levels. We call these variables, or sometimes you'll hear them called controlled conditions, because their values can change. For cells to function normally, each variable must be maintained within a normal range, usually centered around an ideal set point. For example, average human body temperature is often cited as 98.6 degrees Fahrenheit or 37 degrees Celsius. In reality, your temperature fluctuates slightly around this value across the day, rarely more than about 1 degree Fahrenheit in either direction in a healthy person, but it still fluctuates. As long as temperature stays within that normal range, homeostasis is maintained and cellular functions proceed smoothly. Think of the thermostat in your home. You might set it to 75 degrees Fahrenheit. The actual temperature cycles a bit above and below that value, triggering the furnace or the air conditioner as needed. The system is stable not because the temperature never changes, but because it never changes too much. Organ systems function as homeostatic partners. No single organ system can maintain homeostasis alone. Instead, multiple organ systems cooperate. For example, the digestive system provides nutrients and water. The respiratory system brings in oxygen and eliminates carbon dioxide. The cardiovascular system transports gases, nutrients, wastes, and signaling molecules. The urinary system regulates fluid volume, electrolyte balance, and waste excretion. And the endocrine and nervous systems coordinate and regulate responses across the body. Think of a patient with chronic kidney disease. The kidneys filter your blood plasma, regulating the volume and the solute concentration of your body's fluids. They also excrete waste from your body. As the filtering rate declines, the kidneys can't effectively regulate fluid volume, electrolytes, or acid-base balance. Fluid overload contributes to high blood pressure and heart failure. Excessive potassium in the blood, known as hyperkalemia, threatens the normal rhythm of the heart. A related increase in the acidity of the body's fluids, called metabolic acidosis, affects bone and muscle tissue. So a primary defect in one organ system destabilizes homeostasis across many variables and controlled conditions. Modern medicine's job is often to understand where homeostasis has failed and how to restore a normal range of values. The body regulates itself and maintains homeostasis using feedback loops. So let's talk about those. Feedback loops are circuits in which the outcome of a process influences the process itself. In physiology, there are two main types: negative feedback and positive feedback. A common misconception is that negative feedback decreases a variable and positive feedback increases it. That is not correct. Both types can be involved when variables rise or fall. The key is this: negative feedback acts to reduce or resist deviations from a set point. Essentially, negative feedback reverses a trend, whether that raises or lowers the value. Positive feedback acts to amplify deviations from a set point. So essentially, positive feedback enhances an existing trend. Before we look at each type, let's define the basic components of a feedback loop. Any homeostatic feedback loop has three essential components. A receptor. You have to monitor the value of variables so that you can detect changes, and that change is called the stimulus. A control center, often in the brain or an endocrine gland. The control center receives input from the receptor, compares the current value to what the desired set point is, and then decides what to do. And then you have an effector. The effector is the tissue or organ that carries out the control center's desired response that adjusts the variable. Again, a stimulus is any change in a variable that triggers the loop. The effector's response either returns the variable toward the set point, which would be negative feedback, or pushes it further from the set point, which would be positive feedback. Having said that, negative feedback is the primary mechanism for maintaining homeostasis. In negative feedback, any deviation from the set point is made smaller or resisted. Once the variable returns to the normal range, the effector's activity diminishes or stops. Let's revisit body temperature as a classic example of a negative feedback loop. First, the receptors. Thermoreceptors in the skin and deeper tissues monitor temperature. Thermo, like a thermostat, temperature. If body temperature rises because it's hot outside, you're exercising, or you have a fever, these thermoreceptors detect that change. Next is the control center. The receptors send these data to a small part of the brain just above the brainstem called the hypothalamus. Actually, come to think of it, some people consider the hypothalamus part of the brainstem, but we'll get into that in a later episode. The hypothalamus compares your current temperature to the temperature's set point, which is your normal body temperature. And then finally, the effectors. If the temperature is too high, the hypothalamus activates effectors such as sweat glands, which function to cool the body, or the blood vessels of the skin, which dilate, bringing more warm blood to the periphery or to the surface, increasing heat loss to the environment. As the body cools and temperature moves back toward the set point, the hypothalamus reduces its stimulatory signals. Sweat production decreases and blood vessels constrict. So the effectors stop when the variable is corrected. That shutting off of the response once the variable is back in range is the hallmark of negative feedback. And the same principle operates when you're cold, but with different effectors: shivering, vasoconstriction of the skin's blood vessels, and behavioral responses like seeking warmth. In a clinical setting, you might see a patient with hypothyroidism. This means an underactive thyroid. The thyroid regulates your metabolism or cellular activity. People with hypothyroidism have a slower metabolism. That means the cells are doing less work, burning fewer calories, and therefore generating less heat. Cellular activity is our main source of body heat. The symptoms are a lower than normal basal body temperature, an intolerance to cold, and a difficult time keeping weight off. The thyroid produces hormones that are among the key determinants of metabolic rate and heat production. In this example, a disruption of endocrine signaling alters the entire temperature regulation loop. Another classic negative feedback system is blood sugar regulation. Much of the carbohydrates we consume are broken down into molecules of glucose. Some of that glucose is dissolved in our blood plasma and is readily available for cells to utilize for energy. This is referred to as our blood glucose level and is measured in two main units: milligrams per deciliter, which is common in the United States, and millimoles per liter, which is used in other countries like the United Kingdom and Canada. Normal fasting levels of blood glucose are around 70 to 100 milligrams per deciliter, which converts to 3.9 to 5.5 millimoles per liter. Okay, let's talk about the feedback loop, regulating blood sugar. After a meal, the concentration of glucose in the blood rises. You absorb the glucose through your digestive tract and it ends up into your blood. And fun fact about glucose, the receptors and control center for blood glucose are actually the same cells. The pancreas, which is a gland behind the stomach, has cells called beta cells that sense the elevated glucose and act as both receptor and control center, secreting the hormone insulin into the blood. And there are multiple effectors for this feedback loop. Insulin promotes glucose uptake by skeletal muscle and adipose tissue and stimulates the liver to store glucose as glycogen. When we do this, blood glucose levels fall back toward the set point, and then insulin secretion decreases. When blood glucose falls, which is typical between meals, different cells of the pancreas called alpha cells secrete the hormone glucagon. Glucagon stimulates glycogen breakdown by the liver, which releases glucose into the blood, raising blood glucose back toward normal. As glucose normalizes, glucagon secretion falls. Both the increase and decrease in glucose are under negative feedback control. The responses always aim to counter the change, not perpetuate it. Oh, I just thought of one more good example: blood pressure. The prefix baro means pressure. So baroreceptors respond to changes in pressure. In the baroreceptor reflex, stretch receptors in arteries of the neck and chest sense changes in blood pressure and relay information to cardiovascular centers in the brainstem. If blood pressure drops, like when you stand up too quickly, the following happens. The fight or flight half of your autonomic nervous system increases, raising heart rate and the heart's strength of contraction, and it causes the blood vessels to constrict. These measures all work to increase blood pressure. Blood pressure moves back toward the set point, and the increased sympathetic drive fades. Again, the pattern is the same: deviation from the set point, detection by receptors, counter response, and cessation of the response as the variable normalizes. Okay, now let's talk about positive feedback, which amplifies change, hopefully carefully. Positive feedback occurs when a response to a stimulus causes a deviation from the set point to become even greater. The system doesn't resist change, it actually accelerates it. I mean, that sounds dangerous, right? And it can be, but in specific situations, positive feedback is useful, especially when a rapid, decisive outcome is needed. Positive feedback loops are typically self-limiting, meaning they end after a specific necessary event occurs, and they're embedded within a larger context of negative feedback systems that keep them controlled. Let's try a simple example. Let's say you blush from embarrassment. Then you notice you're blushing. You get more embarrassed, and you blush even more. The response, which was embarrassment leading to vasodilation in the face, increases the stimulus, which is awareness and social anxiety, which then increases the response, and so on until the situation ends. I mean, that's a simplistic example. So let's try a real physiologic example: blood clotting, also known as coagulation. When blood vessels are damaged, you want coagulation to happen fast and locally. A chemical called thrombin is generated as part of a series of reactions called the coagulation cascade. Thrombin not only converts an inactive protein called fibrinogen to fibrin, but also stimulates the production of even more thrombin by activating upstream components. This positive feedback rapidly amplifies clot formation at the site of injury. So why doesn't your entire vascular system clot? Well, because the system is self-limited. Coagulation factors are consumed locally at the injury site, so they can't keep stimulating their process. Natural anticoagulants and blood flow dilute and remove activated clotting factors away from the injury. And once the breach is sealed and key reactants are depleted or inhibited, the positive feedback loop stops. Here's another classic example: childbirth. Labor is a classic positive feedback sequence. Since this is episode 2, I'll give you a quick cursory description of the anatomy of the uterus, where the fetus lives until birth. The uterus is like an upside-down Erlenmeyer flask, like the ones you see in a chemistry lab. The large part of the flask is the body of the uterus, and the neck of the flask is called the cervix, which literally means neck. Near the end of pregnancy, the growing fetus stretches the uterus, especially the cervix. Stretch receptors send signals to the hypothalamus of the brain. The hypothalamus signals the posterior pituitary gland to release a hormone called oxytocin into the blood. Oxytocin reaches the muscle of the uterus and stimulates stronger uterine contractions. Stronger contractions push the fetus further into the cervix, increasing cervical stretching. Increased stretch stimulates more stretch receptors and feeds back to the hypothalamus to cause even more oxytocin release, intensifying the contractions again. The cycle continues: stretch, oxytocin, contraction, more stretch, until the fetus is delivered, becoming a neonate or newborn. and the stretching stimulus is removed. Once the original stimulus disappears, the loop eventually shuts down. Since oxytocin is a hormone, it takes a little time to clear it from the blood, so the contractions continue for a bit after birth, which is a good thing because it helps to expel the placenta and pinch off the broken blood vessels that resulted from the labor. The hallmark of positive feedback is effectors continue the response beyond the set point until the initiating stimulus is eliminated. Now, sometimes feedback goes wrong. There is also detrimental positive feedback. Most disease states involve failure of negative feedback to maintain homeostasis or accidental activation of harmful positive feedback loops. One example, hemorrhagic shock and the spiral of low blood pressure. Consider severe blood loss from a major trauma. Massive hemorrhage or blood loss causes a sharp drop in blood volume and, in turn, blood pressure. Initially, negative feedback mechanisms kick in, like increased heart rate and vasoconstriction, to maintain perfusion of the tissues. Perfusion means how well the blood is getting into the tissues. But if blood loss is too great, perfusion to the heart muscle itself becomes inadequate. Loss of blood flow to a tissue is called ischemia, and ischemic cardiac muscle can't contract effectively. So the amount of blood the heart can pump falls even further. This leads to even lower blood pressure, which then worsens the ischemia. The heart pumps even less blood, and the pressure drops again. So now instead of stabilizing the variable, the system is caught in a self-propagating downward spiral, a detrimental positive feedback loop. Blood pressure deviates further and further from the set point until the heart can no longer sustain function and the patient dies. Unless you intervene with volume resuscitation, hemorrhage control, and sometimes medications that increase blood pressure. In contrast, after modest blood loss, like donating a pint of blood, negative feedback mechanisms are sufficient. Heart rate increases, vessels constrict appropriately, and blood pressure is restored to the normal range. This contrast illustrates two key principles about homeostasis. One, many diseases result from failure of negative feedback to maintain homeostasis. Two, some positive feedback mechanisms can be harmful, driving the system away from stability instead of toward it. Okay, one last nuance. Homeostasis doesn't mean every variable is always held to the same narrow range in every context. Sometimes temporarily changing the range is beneficial. Exercise is a good example. During vigorous exercise, heart rate increases. The amount of blood the heart can pump in one beat also increases. We call that stroke volume. Blood pressure rises above the resting normal range, respiratory rate and depth increase, and body temperature may rise slightly. These changes are adaptive. They ensure that muscle cells receive more oxygen and nutrients and can remove more carbon dioxide and metabolic waste. If your cardiovascular system refused to let blood pressure rise during intense exercise, you'd faint every time you tried to climb stairs. After exercise, negative feedback systems bring variables back to their resting ranges. Here's another example, fever. During an infection, biochemicals called cytokines can raise the hypothalamic set point for body temperature. You feel cold and start shivering because, relative to the new set point, your current temperature is too low, even though it's higher than normal. Your body works to raise the temperature. This can enhance immune function and inhibit some pathogens. Eventually, when the cytokine level changes and the set point reverts to normal, you experience breaking the fever, sudden sweating and vasodilation to shed excess heat. In both cases, homeostasis is still operating but around a different set point, which is more suitable for whatever that situation is.
AI agents are everywhere, automating tasks and making decisions at machine speed. But agents make mistakes. Just one rogue agent can do big damage before you even notice. Rubrik Agent Cloud is the only platform that helps you monitor agents, set guardrails, and rewind mistakes, so you can unleash agents, not risk. Accelerate your AI transformation at Rubrik.com. That's RUBRIK.com.
All right, let's bring it all together. Life, homeostasis, and clinical reasoning. As this podcast continues on with new episodes, the details will multiply. Specific ion channels, receptor subtypes, signaling pathways, genetic mutations, physiological processes, etc. It's easy to get lost in the weeds. The 6 essential characteristics of life: organization, metabolism, responsiveness, growth, development, and reproduction, give you a big picture framework. They describe what living systems must do to exist. The 6 essential characteristics of life: organization, metabolism, responsiveness, growth, development, and reproduction, give you a big picture framework. They describe what living systems must do to exist. Whereas homeostasis explains how those capabilities are stabilized in the face of constant internal and external change, primarily through negative feedback with carefully deployed positive feedback in special cases. Think about the day that you eventually reach a clinical setting. Here are some situations you might come in contact with. A patient in diabetic ketoacidosis. Think metabolism and homeostasis of glucose and acid-base balance. A child with failure to thrive. Think growth and development and the systems that support them. An elderly patient with heart failure and edema. Think about organization and how impaired cardiac function disrupts volume and pressure homeostasis. A trauma patient in hemorrhagic shock. Recognize the detrimental positive feedback pulling them away from life and the interventions needed to restore negative feedback control. Ultimately, every diagnosis and every treatment is an attempt to protect or restore the essential characteristics of life by reestablishing homeostasis. If you can always tie the details back to these core ideas, the complexity of healthcare becomes much more coherent and a lot more meaningful. Well, that's a lot to take in, and I'm so glad you've made it to episode 2 with me. There is a lot more to come, and hopefully this podcast will help you get your B or better in A&P, or simply help you understand our bodies a little bit better than you did before. If you have a question for me, please email me at minus55media@gmail.com. Maybe I'll answer your question in a future episode. Once again, thanks for listening, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media and is hosted by the Believe Network. Please take the time to rate the podcast and leave a review. You can also check out the content I post on Instagram and YouTube as @StudentHelp4AP. That's StudentHelp, the number 4, AP. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where your instructor can find Anatomy and Physiology Digital Suite, my low-cost, tutor video-based digital learning solution for anatomy and physiology currently being used at several colleges and universities. If your instructor isn't using Digital Suite, let them know about it. The link is in the show notes.
If you like the show, please take a moment to rate, review, and subscribe. It really does help the show to grow. Thank you for listening. Happy holidays! Want to give your host a gift? Consider subscribing, rating, and reviewing the show this holiday season. It really helps the show grow. From all of us at Believe, have a Merry Christmas, everyone, and a happy holiday.